Hi everyone and welcome back to the Career Discovery Sessions. This week we have an international speaker, a science researcher from Munich who focuses on the research of zebrafish in regards to studying a various range of diseases. Please welcome Krasimir Slanchev.
Thanks once again for inviting me for this interview. I think it's a very pleasure to be here and to talk about my career. So you want to know a little more about what research is like, what science is like. So I'm working at the Max-Planck Institute. This is a scientific organization that is funded majorly by public funds, so it's paid by taxpayers. We do basic research, which means we do any research that can or cannot be applied practically, which means you don't care if a certain research topic is really applicable at the moment, but it could become important in 10 years. But we want to know about the things. We want to know how the things work, how the molecular mechanisms are employed by different in different processes. So research, basic research is practically about doing research for its own and accumulating knowledge that can become useful at some point. In particular, in our lab, we are doing neurobiology research. We work on zebrafish. These are small tropical fish. They have a lot of advantages. They are vertebrate system. They have transparent larvae, and it's easy to look into their brains directly. They are also complex enough to have enough neurons to have complicated behaviors. And this is all advantage when you do research on a small animal. And we are trying to still, they are enough complicated to be related to processes that can be also that the same processes in complicated brains like the human brain. So whatever we learn in zebrafish on for how the brain works, how the neurons are connected, how they function, this all can be applied to humans also and to other species. So we are researching actually on the basic principles of the neurobiology. So in this video, you can see the development of a single zebrafish embryo. It starts with a single cell, which is fertilized, and then it starts dividing. All the cells are dividing all the time, and until you see those small dots, these are single cells. Then at some point around 6 hours, you can see the counter down there. Around 6 hours, it starts gastrulating, which means the cells start moving around the embryo. After 9 hours, they kind of cover the whole embryo, and then starts the gastrulation. You can see already how the small fish is formed. You see the head, you see the eyes, then you see the muscles forming. There is a tail stretching. And then at some point around 24 hours, you can see already that this is a fully developed embryo. It still has a lot of yolk, so it doesn't need to eat yet, but it already has a heart that starts beating. You can see the blood flowing. You can see those small black dots. These are pigment cells, which are also developing. Some of them are migrating as well. And then around 2 days already, the larva gets already big enough and it starts hatching. So it gets out of the chorion and starts swimming around. And then at 5 days, it can start already feeding. It starts hunting. So this is a remarkable thing that you can see within 5 days, you get a fully developed organism that is capable of doing everything. It can swim, it can feed, it can hunt, it can perform complicated behaviors. And all this is done in a small organism that can be fully imaged by the microscope, and you can observe all those processes. So this is very fascinating. And that's actually what captured me in this field. The developmental biology is how you see the integration of all biological processes that come together to form an organism that can function, that can perform all these complicated behaviors and complicated processes.
And which aspect of this time frame would you be researching? Would it be from start to finish, or would you focus on one time frame in particular?
So the nervous system, like in human embryos or in other embryos, in vertebrate embryos, the brain starts developing around day 2. So initially starts, of course, forming around day 1, and then at day 2, more and more cells mature, and the neurons starts forming protrusions, forming contacts. They extend protrusions. They form different areas and specialize further and further. So depending on the question that you're asking, what you're interested in, are you interested in how the neurons develop? Then you can study from day 1 to day 5. If you're interested on how the neurons function when they're already formed, then you can go around day 5, and then you can see how the neurons, actually we have those tools where you can make the neurons, you can visualize actually when the neurons get activated. So in fact, you can see in live how the neurons uh, are firing up. This is the second video that I gave you. I don't know if you can also put it on the screen if you want, but this blinking, this firing up, these are single neurons. The fish is just sitting there. It gets different cues, different information from the surroundings, and then it starts analyzing this information, and you can see which parts of the brain are actually analyzing at this moment. For example, if the fish sees a specific thing, then a certain area of the brain lights up, which means the fish exactly. So you can practically observe the thoughts of a small zebrafish embryo. And depends on what you're interested in, what process exactly you study. You can go into different stages of the larva and study those.
Great. And what about the other diseases that you study in your research? How do you observe those in?
Um, what
Okay.
what you mean?
So I know you sent me the paragraph where you said you study diseases through zebrafish.
Ah, okay.
So apart from the neuroscience aspect of it, what else can be observed
Mhm.
from?
So, for example, at the University Clinicum in Freiburg, where we studied kidney development and disease, we were able to study proteins that are involved in the formation of cysts in human kidneys. So we can take the same genes, the same proteins in zebrafish, we can mutate them, or we can knock them out, we can turn them off, and then we can see what happens to the fish kidneys, because the fish have kidneys. I mean, this is not very well known, but they actually extract water all the time because they live in water. So they have very active kidneys, and their kidneys look very similar to a single human nephron. So this is the unit of a human kidney. And using zebrafish as a model, we could actually confirm several disease, like several genes that have been suggested to cause a certain disease in humans. Those are usually so-called celiopathies, and they involve the movement of a small cilia in the kidneys, which is driving the fluid out of the kidneys. And if you mutate certain proteins, you can see that those cilia are not moving anymore. And that's how you can actually prove a causality, a causal effect, that you suspect that certain gene is important for the disease that is found in human patients. And then if you go in zebrafish and you knock out this gene and you see the same process happening in zebrafish, because they form also cysts, they form malform their kidneys, then this is a proof that this gene is definitely the one that you're looking for. And based on that, you can look for therapies. Of course, you can say, okay, how can we influence this gene? And then you think about medications or therapies or different ways to cure disease. You can apply this aspect to all systems in zebrafish. You can study the skin, you can study regeneration, you can study wound healing, you can study kidney development, you can study eye development, neuronal development, whatever you can think of.
Great. So it's a really elaborate field of study.
It is an interesting field.
Research is quite a long-term project, but what is it that you do on a day-to-day basis?
Well, the daily routine of every scientist can be quite different. So you get up in the morning, you have breakfast, you send the kids to school, and then you go to the lab and you do whatever is needed to be done. This can be someday it can be reading papers, another day it can be writing proposals for grants, another day it could be doing experiments with animals, another day it can be analysis of the data that you collected from those experiments. So the research daily life can be quite variable, which is a good thing. You don't get into routine. It's also relatively flexible because you can depending on the deadlines that you have and on what you have to do. Actually, you can choose when to do it and how to do it. So it's interesting. I like it. It's fun.
And is there a negative aspect of it?
Yes, of course, as every job, there are negative aspects. So research is very competitive. There are multiple labs in the world that are working on similar topics, and only the first to discover a certain thing gets to publish it and gets to achieve something. So there's a lot of competition. There is stress. There are deadlines. There are also negative aspects. Yes.
And what about the skills required in the office?
Most of the time, the skills that are needed are you have to have logical thinking. So you have to be able to plan experiments. You have to be organized. You have to have basic knowledge on the things that you do, and not only basic, but also like deep knowledge. You have to keep up to date with the current literature, with the newest finding in your field. You have to you have to have good communicational skills, which are not, which you don't see in all researchers, unfortunately. You have to be able to present your work. You have to be able to summarize your work. You have to be able to plan in the future. So pretty much those are skills that are not only research relevant, but they are connected to every other business, I would say.
I would also like to focus on the path to get to where you are now. Could you tell us how you reached this position?
So I began my studies in Bulgaria, the University of Sofia, and I studied there at the biological faculty. Then I graduated with a Master of Science degree there in molecular biology. Then I worked for 2 years in a chemical lab, chemistry lab in at the Bulgarian Academy of Sciences. And then I got accepted in one of the Max-Planck International Research Schools in Göttingen, and I came to Germany. In Germany, I did from the research school, I got my another master degree in developmental biology, and then a PhD degree again in developmental biology. After I got my PhD degrees, I started working as a research fellow in different organizations. This was in Freiburg at Max Planck Institute, and also at um, the Clinicum, University Clinicum in Freiburg. And eventually I got a staff scientist position at this Max Planck Institute in Munich, and I moved here. So it was a long trip, but that's how the research career is.
Right. So there's quite a lot of requirements to become a researcher. Yes.
Yes, sure. I mean, there's a lot of study.
Are there any similar career paths in your field?
There are not, it's pretty much similar for everybody who wants to do academic career. It starts with university, and then it goes on with a master, PhD, and then eventually professorship, group leader, professorship to get to the top. However, there are a lot of students that start this path, and eventually some of them deviate during the course of studies. So after you get your master degree, you can decide to quit research and you want to go into the some companies or spin-offs. The same can happen after you finish your PhD or at later stages. So there are a lot of paths that lead from the research initially.
And you speak of all these later activities they build up on each other. But do you think there are any activities that a student of my age, 16 or 17 years old, can do, like work experiences or trips?
it's such a dynamic field that every month or every week there are new findings published. So only reading only textbooks is not enough. You have to kind of so once you read the textbooks, you have to keep on catching up with reading the newest published papers and reviews and articles. So I I don't have a particular recommendation. I mean, whatever is interesting, you can read it now. It's in any case, I would encourage you to read mostly about what you're interested in, what is your main feeling that what you feel good in reading about, and what you find interesting in doing, and that's actually I think more important than reading a particular textbook at this age.
Can you also speak a bit about similar careers? Who do you interact with in your lab?
So research is a little bit like a bubble. We are quite isolated, and we don't. I mean, we communicate. We of course we interact with many other researchers and also with other fields, but the academic career does not necessarily involve interaction with people from other fields. For example, I know very few Hollywood stars and also very few I don't know, millionaires. But I mean, of course, we meet colleagues or people that have followed, at least in the beginning, a research path, and then they ended up working for a company or decided to switch to a pharmaceutical company or to other production field. We communicate with those. In general, the academic pathway is always the same, and you follow it, and at some point you deviate from it. And of course, it's interesting to know what other people have, how other people have achieved it. But yeah.
And finally, I'd like to ask, do you have any motivational advice for all of our students who are listening and might consider this a career choice in the future?
Motivational. Yeah, I mean, you should follow up what you're interested in. I think this is the most important thing. I guess it's hard. It's getting increasingly hard to predict what will be hot or what will be interesting in the next 10 years or 20 years. There are no promising professions. I mean, of course, there are some fields that you have to always follow, but um, I would say follow your own interests and become good at this. And when you're good at this, when you're good at something, then there will be always an opening for you. So there will be an opportunity for you, I think.
Actually, I'm going to override myself here. One final question. Do you
Okay.
think your field is growing based on what you said?
Research field, definitely. I mean, this is you see that there is a huge expansion of the research, and especially the life science research. Even with this pandemic now that occurred, even the COVID pandemic, suddenly everybody speaks about the research, everybody speaks about science. PCR, who knew what PCR was like a year ago? I mean, we were using that all the time, but no one from the public knew about it. And now everybody speaks about PCR and about spikes on viral uh, on viruses and immunization. And so research is an increasing field. It will get even more important because we are getting deeper and deeper in the knowledge of how things work. So our life is getting more and more complicated. So definitely the research field is expanding.
Thank you so much. This has been really helpful for all of the students.
You're welcome. I hope it was helpful.
I hope you gained some insight into the day-to-day life of a researcher for science and learned more about the zebrafish study, which is a very interesting and surprisingly large field that compares and influences the human world. See you next week when we're going to be talking to a completely different individual. Bye.